I actually feel that it's probably not optimal to be on a ketogenic diet all the time. I follow a modified version of it that's, very rich in plant-based, fiber and things like that. Um, so for the average person, there are definitely benefits of intermittent ketosis. And I think the advantage of going into ketosis occasionally is that it promotes metabolic flexibility and opens up and really pushes your body to be more fat adapted.
Hi everyone, welcome to Live Longer World. I am your host Astha, and in this podcast I speak with longevity scientists working to slow and reverse aging. To find all podcast episodes and other resources, go to my website livelongerworld.com. To find the link to the show notes for this episode, check out the description below. I have a very special guest today, Dr. Dom D'Agostino, who needs no introduction in the world of ketogenic diets, but here's a super brief one. He is an associate professor with his research focusing on metabolic-based therapies, ketogenic diets, ketogenic supplements, and other metabolic-based drugs. Make sure to check out his website, ketonutrition.org, where he educates people like us and other consumers on his research. Okay, now let's dive into the episode. Hi, Dom, and welcome to the Live Longer World podcast.
Great to be here. Thank you for having me.
So, firstly, I want to thank you because you're a huge part of the reason I started practicing ketogenic diets myself. And this was about 2 years ago. Now I do mostly a modified Atkins sort of diet. I think similar to what you do because I incorporate a lot of vegetables, especially cruciferous vegetables in my diet. But I feel so good. I absolutely love the diet. I love the brain clarity benefits and just overall energy and lightness. So thank you for spreading the word on it.
Great. Well, yeah, thank you for listening to, whatever information you heard or read or whatever, and advancing sort of the message behind here. I think not a ketogenic diet is not for everybody, but I think it is important to reduce the amount of sugar and processed carbohydrates that we're consuming. And I'm actually a fan of a higher plant-based, higher veggie, incorporating veggies into the diet. Actually, the first blog that I ever did on keto nutrition, if you go way back, the title of it is Implementing a Plant-Based Ketogenic Diet, because I get so many questions about that. And I personally felt that there was a lot of benefits that people can derive by maybe reducing meat and incorporating more plants in that. So actually, I made that the first blog that kicked off our website, blog. That was the first blog ever. So way back, it was like 4 or 5 years ago.
Yeah, and that's certainly possible. I mean, I'm vegetarian actually. I don't eat any meat, so it's totally possible. And we'll dive all into that and a bunch of questions that you get asked on it. But I think I want to start with just the history of ketogenic diets, because most people, they may have heard of keto, and it's a popular diet. It's often synonymous with just a weight loss diet, but I think people don't realize that, it actually started as research for neurodegenerative diseases or for neuroprotective against seizures and epilepsy. And a huge part of your research is focused on that, in fact. So maybe you can touch a bit about the connection between ketogenic diets and protecting the brain against neurological disorders.
Yeah, thanks for bringing that up. The history is really important, and I was in a dietetics program at Rutgers University for my undergrad, and later majored in bio too. But during my training in nutrition science and dietetics, we didn't really talk about the ketogenic diet, just that it was a bad thing to do, to even think about. We didn't talk about the clinical use of it. We did talk about protein-sparing modified fasts and how people died from doing, this zero carb, high protein diet, which was just basically drinking collagen. They became protein deficient. That's kind of another story, but the ketogenic diet has its roots at the Mayo Clinic. It is the 100-year anniversary of the ketogenic diet, actually, and we just got our book, which kind of commemorates that, with some of the leaders at Johns Hopkins, Dr. Eric Kossoff, as part of this, Dr. Jung Roh, who is at Barrow Neurological Institute. He's at UCSD now. They got me interested in the ketogenic diet. Dr. Eric Kossoff's mentor was the late John Freeman, who really advanced the use of the ketogenic diet in pediatric epilepsy at Johns Hopkins. Prior to that, work was done way back in the 1920s at the Mayo Clinic, and they designed a diet that was very high in fat, up to over 90% fat. And the idea was to mimic fasting, because we knew through centuries or millennia that fasting was a way to control seizures. And if you fasted, you had the presence of these things in the blood and urine called ketone bodies. And one way to mimic the metabolic physiology that would be associated with being in the state of ketosis was to restrict protein. Many people don't know the ketogenic diet is a protein restricted diet. So to restrict protein and carbohydrates, probably the most important part of being in ketosis, but it's protein restricted and it's just primarily fat. The protein is titrated to prevent protein malnutrition, and it's low enough to prevent gluconeogenesis. So by eating very high fat, adequate protein, and essentially zero carbohydrates, if you were to draw blood off a person eating this diet, it would look like they're fasting. So insulin would be very low, IGF-1 would be low, mTOR, things associated with longevity, and we could talk about that, activates and increases NAD plus levels too, which is kind of interesting. But it elevates beta hydroxybutyrate, acetoacetate, and also with spontaneous decarboxylation, you get breath acetone, like a fruity smell. And it was the physiological state of being in a state of ketosis, which had the anti-seizure effect. And prior to anti-seizure drugs, this was the standard of care for epilepsy and other seizure disorders. And then drugs came along in the late 50s and 60s and 70s and largely replaced the ketogenic diet. And then there was a resurgence in 1996, I believe, and it was spearheaded by by Jim Abrams, and he was working with Dr. John Freeman, and his son Charlie was put on a ketogenic diet. And Jim, I know Jim well, and he had connections in Hollywood, and Meryl Streep did a movie about the ketogenic diet called First Do No Harm. And it was on a show that I watched back when I was in college, actually studying nutrition at the time, called Dateline NBC. And maybe it's still on. It was on the NBC network. And I found it kind of interesting, but it didn't appeal to me so much that I wanted to look into it further. But the ketogenic diet was used as, it was the only thing that worked for Charlie to control his seizures. And he was on many seizure medications. He got off the medication, started the diet, and it cured his seizures. And what was interesting to me is that when you go on anti-seizure medication and then go off of the medication, you get rebound seizures. Whereas with a ketogenic diet, Charlie was on the diet and then came off of it and never got seizures again. So it cured his diet. There was about 15% of people are super responders, which means they have rapid and permanent loss of seizures. They never get seizures. So they have to follow the diet for a period of time. And then after a period of time where they don't have seizures, they come off of it gradually, and then they never have seizures again. And then the etiology of epilepsy is largely unknown, and it's caused by a variety of different things. Some people go through puberty and have seizures, and there's many different reasons why people have seizures. But the ketogenic diet tends to work for many different seizure types independent of the etiology. So you can have, absence seizures or temporal lobe seizures, and tonic-clonic, what we call grand mal seizures, Lennox-Gastaut syndrome, Dravet syndrome, Rett syndrome. There's a variety of, and we study oxygen toxicity seizures, which are super powerful tonic-clonic seizures. And then, fast forwarding, we tried it for that, and it was remarkably effective for that. So I became very interested scientifically as to the mechanism, how are ketones, how are being in a state of therapeutic ketosis, how does it have a neuroprotective effect? And that I got talked into this area of research when I was studying drugs, and then transitioned to more dietary or supplement. We developed exogenous ketones too to do that. And that was about 2008 or 9. And that changed the trajectory of my career from drug-based research to more nutrition and supplement research.
Well, that's exactly what I was going to ask you next. What is the mechanism by which these ketone bodies might be anti-seizure or seizure protective?
Yeah, that's an area of intense investigation. And the thick book that I just showed you, there's many chapters in there from top tier researchers at, Ivy League at Harvard and Yale and Duke and Johns Hopkins and Mayo Clinic that have written chapters about the mechanism. What the belief is that the general consensus now, it wasn't maybe back when I started studying this, but it's very clear that it's multifactorial. So the ketogenic diet works through many mechanisms working in synergy. And it tends to work when ketogenic or when anti-epileptic drugs fail, and even multiple drugs. They may work in the beginning, but they fail. Many drugs fail. About a third of the patients are completely drug refractory. And you can put them on a ketogenic diet, and more than 2/3 of them respond very favorably. And some of them respond remarkably well, super responders. So we kind of had the understanding that it was working through a mechanism independent of what any drug was working through. So what we study in our lab is a variety of different mechanisms we look at. The thing that got my attention was a reduction, and I saw this in cultured neurons, hippocampal neurons and cortex neurons, and also in hippocampal brain slices, which I did my PhD as a patch clamp electrophysiologist. Maybe people listening to this may not know what patch clamping is, but it's you take an electrode and it's a microelectrode, and you can basically seal on to a neuron in the brain or in culture, and you create like a giga-ohm seal. And then it's more of a biophysics technique. And then you can measure action potentials and membrane potential and firing frequency, and determine what channels are being activated and things like that. So doing a variety of techniques, we demonstrated that reactive oxygen species go down, in particular superoxide anion. And I was using a method using dihydroethidium to measure that. And it was a remarkable reduction. And in neurons in culture, we could throw a variety of different toxins at them, including high pressure oxygen. And oxygen is toxic at a certain level. And then the neurons seem to maintain their membrane potential and firing frequency. And there was a pretty sharp reduction in oxygen-free radicals when in the presence of ketones, beta-hydroxybutyrate and acetoacetate. And I thought this was very important and very relevant to the work I was doing in oxygen toxicity, seizures, which is the limitation for Navy SEAL divers, also for hyperbaric oxygen. It limits hyperbaric oxygen. So, and it was doing it through a way by enhancing mitochondrial function. So if you give a metabolic fuel, neurons can run off glucose or run off ketones if you're fasting, and maybe lactate too. So definitely lactate, but generally speaking, your brain runs off glucose like 100% on a standard American diet. And then if you stop eating today, by this time next week, your brain would be using about 60% of its cerebral energy metabolism will be derived from beta-hydroxybutyrate and acetoacetate, so the ketone bodies in circulation. And this seems to provide an energetic advantage to the brain under some circumstances, in particular seizures, and in the presence of toxins. So like oxygen, or I was using like glutamate and various things to cause oxidative stress and excitotoxicity. And so I became very interested in ketones as a neuroprotective compound and an anti-seizure compound. So the work in cell cultures got me to write different grants, and then convince the Department of Defense to study this in a rat model. And in a rat model, the use of previous work by the military did fasting but you can't get Navy SEALs to fast for days prior to going into a mission. So the idea was to mimic the metabolic physiology of fasting by administering a ketogenic compound. And in our case, we administered a ketogenic compound 30 minutes prior to a simulation of a high pressure oxygen exposure. And in this case, it had a remarkable effect at delaying the latency to seizure, which we actually do. We measure EEG, we measure diaphragmatic EMG, and we have body temperature. We measure many different variables under these conditions, in addition to having a video camera system set up to show the behavioral seizures. And under these conditions, it was far more effective than any anticonvulsant drug that was ever tried, by administering these, not all compounds. So the first ones we tested didn't have any effect, and I started to lose enthusiasm. And then we developed and tested a compound that elevated beta-hydroxybutyrate and acetoacetate in a 1-to-1 ratio. And we can talk about the redox effect that this had. And it seemed most important not to elevate beta-hydroxybutyrate, but to elevate beta-hydroxybutyrate and acetoacetate sort of in a certain ratio. And it was not totally clear at the time, and it's still not totally clear why that's the case. But experimentally, we saw this in different animal models. My colleagues were sending me papers, and it was, so I worked with a professor at Case Western, Dr. Henri Brunengraber, who chaired the NIH-sponsored metabolomics core at Case Western, and he gave me the recipe, more or less, to synthesize a ketone ester, a particular ketone ester that ended up being very efficacious in this animal model of oxygen toxicity. And then later, we used this particular ketone ester in a variety of different disease states, including cancer, including Angelman syndrome, other types of seizure disorders. And then we developed ketone electrolyte salts, which are the ketone body beta-hydroxybutyrate, which has interesting effects at sirtuin activation, BDNF, and things like that we're studying. But beta-hydroxybutyrate bound to like sodium, potassium, magnesium. So an electrolyte that you can consume that tastes good. So that was a big advancement. And actually you can elevate ketones and with something that actually tastes good and delivers electrolytes too, which are kind of important under some circumstances. So like these are the things that I consume on a daily basis and that are commercially available now. And then we're using them in similar studies and getting very interesting results with these compounds. So, I mean, we have a library of like 60 or 70 different compounds. So there's a variety of different ketogenic agents that you can develop, including things that are fats, like medium chain triglycerides are a type of fat that you can consume. And then they go to the liver via hepatic portal circulation. They're not packaged into chylomicrons like long chain fats. And then through beta oxidation in the liver, it generates a lot of acetyl-CoA, and this condenses to acetoacetate, and then it stimulates the ketogenesis process. So you can sort of naturally coax your body into producing ketones through these ketogenic fats. And what we find when we combine exogenous ketones with these ketogenic fats, it seems to further augment the therapeutic efficacy of these compounds. So there becomes an interesting, experiments of formulating different compounds for different disorders. So we're really in the process of that. And then my PhD student, I just finished a long meeting with her, and she's extracting histones, and we're looking at DNA methylation and then histone modifications that are driven by beta-hydroxybutyrate. So beta-hydroxybutyrate can affect histone deacetylase enzymes, but beta-hydroxybutyrate can also directly interact with histones through a process called beta-hydroxybutyrylation. And something happens similar with lactate. But the beta-hydroxybutyrate can directly interact with the histone and cause modifications that have a favorable effect in the disease processes that we're studying now.
That's fascinating. Lots going on. Well, one question I think that comes up commonly is that, okay, sure, it can be protective against people who have epilepsy, for example. But if for someone who's healthy, it's like anyone who's practiced the ketogenic diet, they know the mental clarity benefits that you get.
but people have this notion that you shouldn't somehow be doing this on a consistent long-term basis. For example, when I told my friend who was a doctor that I'd be practicing this, she said, no, your brain absolutely needs glucose to run. It can't survive without it. So you
Yeah.
shouldn't be doing this for maybe after your experimentation is done. So what research do you have on, how it could possibly be beneficial for healthy people as well, in terms of the ketone bodies for the brain specifically?
Yeah, even in this book here and in the book by the Hopkins group, I think like even in the introduction, it says the ketogenic diet is not a natural diet. It's an unnatural state, and it shouldn't be followed, and it should be thought of as a medical therapy. It's a very calculated diet. It's the only diet that we know of that's defined by an objective biomarker that you can measure, right? So like there's like the carnivore diet, paleo diet, all these diets. But the ketogenic diet is a very scientific diet, a lot of research behind it. To achieve therapeutic ketosis involves a pretty significant rearrangement of the macronutrient ratios. So the classical ketogenic diet being like upwards of like 85 to 90% fat, and then just enough protein to prevent malnutrition and essentially zero carbohydrates. Then you have modified versions of the ketogenic diet. So one can, and I actually feel that it's probably not optimal to be on a ketogenic diet all the time. I follow a modified version of it that's, very rich in plant-based, fiber and things like that. So for the average person, there are definitely benefits of intermittent ketosis. And I think the advantage of going into ketosis occasionally is that it promotes metabolic flexibility and opens up and really pushes your body to be more fat adapted over time. And a lot of people use this term very loosely, and it's very kind of nebulous and unclear to many people actually what this is. But I mean, yeah, you can measure your respiratory quotient and measure fat oxidation, relative to carbohydrate oxidation, and achieve that. But I would say it's very unnatural to follow a standard American diet and never enter a state of ketosis. So, our history, we had very limited food availability. So we'd probably go through periods of fasting, but also very limited availability of carbohydrates, especially sugar and processed carbohydrate. No one would debate that. But I think with the combination of limited carbohydrates availability and limited food availability overall, one could make the statement that it would be very unnatural to not go in a state of ketosis periodically, even throughout a time frame of 1 month. So something like a fasting mimicking diet advanced by Dr. Valter Longo. I kind of choose to fast maybe 3 days every couple months. And that gets you into deep ketosis where your ketone levels are above or beyond your glucose levels. And when you achieve this and you try to measure insulin, it's almost non-detectable. IGF-1 goes down. You're probably activating a lot of autophagy. Although, I mean, we're doing this in mice, looking at LC3 and other things. And I think if you look at the mouse data and you achieve a glucose ketone index of 1, where glucose bottoms out and then ketones in millimolar concentration get up to like 3 millimolar, and a mouse glucose is like 6 or 7, and that drops down to 3. And then you take a sample of the muscle or liver, then you have signs of autophagy. So, whether that translates to humans, I don't know. I don't want to go on the record of saying that because we need to do these studies, but I'm willing to start doing this before the human studies are done, and occasionally do a 3-day fast after about 48 hours. Then you really dig into, start liberating a lot of fat, and you start getting into ketosis, and then insulin becomes very low. So, and I think that can offer, longevity advantages. And no doubt there are, like Dr. Valter Longo has kind of demonstrated in his studies, doing a fasting mimicking diet, you can do that, and then you have benefits that last for weeks, after just doing this for 5 days. And similarly, I think if you do like a 3-day fast, I think you can get benefits for months even after that. Sounds a little bit far-fetched, but I'm starting to believe this now. And I think it's a good exercise for your psychology. I think it gives you a sense of control. I think it changes your relationship with food, and I think that's a whole nother story that we can talk about. But I think it's multifactorial, the benefits you can derive from fasting periodically.
I couldn't agree more. I've tried them a few times and had the same experience. So on the point of, I guess, doing it more intermittently, I think Dr. Eric Verdin also had a study where he did this cyclic ketogenic diet in mice. I think it was 1 week
Mhm.
on, 1 week off, and it improved memory or something in old mice. Do we know whether, it might be better to just, eat regularly healthy, what you're doing, and do, say, a 3-day fast and get into deep ketosis versus if I don't do this 3-day fast and I'm just in doing a modified keto diet the whole time? Do we have any research on what the pros and cons of that would be?
Not human research. No,
Yeah,
we're
mice
kind of
research,
interested in.
your hypotheses.
Yeah. Well, I mean, if you're asking like what the optimal diet is, that's very individual, depends on many different factors, right? And we have to take measurements, right? What gets measured gets managed. So there are many biomarkers. We can talk about that. But generally speaking, like a lower carbohydrate diet, 100 grams or lower, I think of it as the cornerstone of a diet is protein. Now, protein can be sort of work against you from a longevity perspective, but and that needs to be moderated. But if you control for calories, I think a more moderate to higher protein is probably okay, as long as your calories are not like you're getting surplus amount of calories, excess protein. So adequate protein to ensure muscle and repair, and then a lot of healthy fats, monounsaturated fatty acids, omega-3s, MCTs, things like that. And then I think of carbohydrates in the form of fiber, like cruciferous vegetables, some berries. I have dark chocolate every day, and but it's all moderate. You can do like 50 to 100 grams of carbs a day, and actually get into a state of ketosis. And then periodically, unless you are using ketogenic diet to manage a chronic condition, and there's a variety of conditions that are responsive to a ketogenic diet. But I think it also comes down to calories, and even many of the benefits of a ketogenic diet for weight loss, type 2 diabetes, and things like that really come down to caloric state. And a lot of people don't want to hear this, but from weight loss perspective, there's nothing magical about the ketogenic diet. From reducing hyperglycemia, there's really nothing magical. I think you can reduce insulin and glucose with a diet that's high in carbohydrates. It'd be beneficial if you had higher fiber, and it would probably be similar to a ketogenic diet. But there are benefits to a ketogenic diet on glycemic variability. I mean, I see this. I wear a continuous glucose monitor, and independent of calories, there's less glycemic fluctuations, and I think that could be beneficial for appetite control, and insulin stays a bit lower, at least postprandially, with that. So, I think I'm not really answering your question, but I'm just giving you some ideas if people are looking for like that optimal diet, and if the ketogenic diet is right for them. I encourage people to try it. And one thing that I encourage people to do is to track. And there's 3 different ketone bodies. There's blood. There's beta hydroxybutyrate, acetoacetate, and acetone. And that's measured in blood, urine, and breath, respectively. So the Abbott Precision Extra or Keto Mojo device is good for blood. There's a variety of keto sticks and Siemens multi-stick SG, I think we use. It also measures like pH and specific gravity and things like that. And then there's a breath ketone meter that I use. There's a variety on the market. The BioSense device is more of like used clinically, FDA approved device. it measures breath acetone. And breath acetone is a very good correlate of fat oxidation and seems to be, I was interested in breath acetone because it correlates very well with seizure control. So when parents have a child or someone has epilepsy, I actually tend to, only over the last couple of years am I recommending breath acetone now, because if acetone's elevated, you're definitely in a state of ketosis. The devices are now very quantitative, and you don't have to prick your finger. You can measure many times throughout the day, and you can blow in as many times as you want, and it measures it, and it's very accurate. So so these devices it's kind of like my preferred way of measuring ketosis.
Okay, amazing. Well, I want to talk a bit about ketogenic diets for longevity, and you already mentioned that you see some of the benefits, say, that mimic fasting or even mTOR inhibition that you mentioned. One question there is that, do you see these benefits even without being calorically restricted? Is it something about the ketone bodies itself that's perhaps causing, say, inhibition of mTOR and other benefits that you see?
So for epilepsy and for seizures and neuroprotection and cancer and things like that, yes. So it took me a while to really believe that, though, because my entrance into the ketogenic diet was actually following the caloric restriction data. Mark Madson's work comes to mind. I was researching fasting, calorie restriction, and then I got turned on to the ketogenic diet, and I was like, well, all these benefits are probably coming through caloric restriction. And some of them undoubtedly are, especially, weight loss. It has to be. I think Atkins used to promote weight loss without having to count calories. It's all the magic of the diet does it. But no, you are inadvertently, whether you're trying to do it or not, you're inadvertently producing a calorie deficit when you follow a ketogenic diet, probably because it's hyper satiating and hypopalatable, right? So the protein and the fat tend to have a satiating effect on our brains. And then it's not the hedonic response that we get from fat and sugar together causes us to eat more. So we tend to get surplus amount of calories, and that's virtually abolished with the ketogenic diet. So, but for epilepsy, the ketogenic diet works better if it's calorie restricted, but does not need to be calorie restricted. So with kids, actually, you can bump up the calories on a ketogenic diet to get them to gain weight, and you still have pretty good seizure control for that. But in some kids, though, you get less seizure control if the calories get too high. So and then also, if you get surplus amount of calories with such a high fat diet, triglycerides go up, and you don't want that. So generally, you just titrate the calories to meet the calorie needs and just try to prevent growth restriction with kids. But with adults, you can get seizure control with a eucaloric diet, without weight loss. Now with cancer, my colleague, Dr. Thomas Seyfried, was very adamant that the ketogenic diet need to be calorie restricted to really suppress IGF-1, mTOR, activate AMP kinase, and really drive down the insulin-induced, signaling and glycolysis. So we started doing some research, and calorie restriction is kind of difficult with mice because you have to individually house them, and then you give them the little 5 to 6 grams of food, and then they eat it within an hour, and then they fast for 23 hours, and they eat it, and then so it becomes more like time restricted feeding. So we just gave the ketogenic diet unrestricted, and I developed a ketogenic diet, that was a little bit more favorable. It had like flaxseed oil, MCT oil, and some other, a high percentage of MCT to get them into ketosis, because it's kind of hard to get mice into ketosis. And then we gave this unrestricted ad libitum. And then we implanted, very highly aggressive metastatic cancer cells. And we did a pilot study. And then after like 3 weeks, pretty much all the mice on a standard diet had like rapidly growing tumors, and they died of tumor burden, and pretty much all except for one of the ketogenic diet mice were still alive. So I was like, wow, there's something to this, and we're not restricting the calories at all.
And they were eating, both groups were eating the same calories?
Well, good question. The ketogenic diet group was a little bit lower body weight. So then we did, this was the pilot, and then I had a graduate student at the time, now Dr. Angela Poff. She was like, I want to make this my dissertation. So we got more animals, and then we divided the groups, and we gave ad libitum standard rodent chow and ad libitum ketogenic diet. We call it the KD2 because it was like a modified. It was higher in protein, actually, like 20% protein instead of 10. And so that was a big change in the ketogenic diet during the time, because 20% is kind of high when we're talking about ketogenic, not high for the standard American diet, but kind of high for the ketogenic diet. But we saw a significant increase in longevity of these mice that had cancer, and then we coupled it with hyperbaric oxygen therapy, which is another thing, and they lasted longer, and we published a few papers on this. But at first, the reviewers wanted us, and they had for good reason. They said, well, we think you're seeing a caloric restriction effect. So what we did is we took the standard diet and calorically restricted it to decrease the body weight by 10%, I think, to match it. So the body weight reduction was matched perfectly. Mm-hmm. And we did with caloric restriction standard diet, I think we saw like maybe a 15% decrease in the tumor burden, but it wasn't like the 60% we saw with the ketogenic diet.
I see.
So this gave us more confidence that, at least in this model system, which was a pretty good model, that it had an anti-tumor effect. And the problem with the ketogenic diet, calorie restricted in cancer patients, is cancer cachexia and weight loss. And that was kind of a big issue that we wanted to get around. So it does appear that in animal model systems and in human patients that they can get enough calories with a ketogenic diet, and you're still getting a tumor suppressing effect. I would say it's taking, there's no cure for cancer, but it does take the foot off the gas pedal. I kind of put it in those terms of cancer growth by suppressing the hormone insulin, and then IGF-1 and mTOR goes down. And in the context of some therapies, the standard of care therapies like chemotherapy, radiation, immune-based therapies, and maybe even metabolic-based drugs like the PI3 kinase inhibitors, which tend to have a counter-regulatory effect of increasing insulin. If you couple that with a ketogenic diet, these drugs work better because it keeps insulin low, and then you can, inhibit that metabolic pathway, and you get better effects.
And have you tried the experiment in healthy mice where you give one group a ketogenic diet without caloric restriction and another group just a regular diet, both without caloric restriction, and if the ketogenic diet has any lifespan benefits?
Yeah, those studies take about 2 to 3 or 4 years. We talked about doing this, and we have so many projects in the pipeline right now. Like, we're always using mice. Like, we had mice set aside for these longevity studies, and like, we'll let them live to a year, and it'll be like a 40-year-old starting a kid, and then we'll see how long. But then we get into these other things where we need mice to test a particular compound, or we need to do this or that. So what I would like to do is get a grant to study that, and where we have like metformin, rapamycin, ketogenic diet, at different types of ketogenic diets, like maybe a lower protein ketogenic diet would be interesting to study versus a higher protein ketogenic diet. So these are all studies like we really wanted to do. And then also the question is, can we put mice on these diets and then give them an agent that would stimulate cancer, like various chemicals in the environment, or we can expose them. There are mice that have no hair, and you can do like UV radiation that could induce like skin cancer and things like that. Can we suppress the formation of spontaneous tumors with a sustained dietary intervention? And this is a really important question, but the federal government, doesn't fund these things, and we need to we need to start doing research, because cancer prevention is like, really key in knocking down cancer rates. And if we could develop cancer prevention protocols, like if you live in a family where like 80% of the people are dying of cancer and they get it, by the age of 60 or 70, and if you could start implementing these things early on in life, like in your 30s, 40s, and 50s, then you could really increase your odds at getting cancer or delaying the onset of it.
Yeah, I would love to see that study done. I mean, if we can have something like a dietary intervention, that could be so useful for so many people, like you said.
Yeah, we're working with different foundations, and I think that have an interest in this. Through a university system, it's a little bit difficult. As a professor, you're really encouraged to get large federal grants that pay full indirects and overhead, things like that. But I mean, we're in touch with groups of people that are interested in funding this, and I'm trying to like maybe fundraise to start a pilot study where if we get some encouraging data, that may be the basis to do a larger study to look at these things. Some of this work has been done, but not in the methodical way that we want to do it, where you have mice that have spontaneous tumors, and then we want to make it as relevant as to the real world scenario as we can, and actually develop a diet that's like a standard American diet, where we actually go to the grocery store and like get foods off the shelf and formulate something with a macronutrient ratio, maybe that have some of these oxidized seed oils in it and high fructose corn syrup and things like that. Would be very interesting to see, because the standard rodent chow is what we compare things against, and that's a pretty healthy diet, relatively speaking. That would be like a healthy version of like an American diet. So, and that's what we're comparing it to. So we think that our data would be more robust if and it may point to the harm that a standard American diet and different types of foods are having.
Absolutely. Yeah. So one thing you mentioned a couple of times was the insulin signaling pathway. And I believe that might also be one of the mechanisms by which, say, ketogenic diets could have longevity benefits, where it just activates that pathway less. I was talking to Dr. Richard Miller on my previous podcast, who runs the interventions testing program for testing different longevity supplements. And he actually spoke about acarbose, which is a drug that's given to diabetic people. And what it does is that it blunts the peak level of glucose. So it's not about just like your moderate levels, it's like the peak level of glucose gets blunted. And that actually has quite good lifespan benefits. When you combine that with rapamycin, in mice, he saw the highest lifespan benefits. In fact, it was for male mice, it was 29%. So I'm curious, maybe that got me thinking, maybe there's some connection here with, just blunting the glucose levels. And ketogenic diets in some ways do the same thing, which could be having longevity benefits and activating the insulin signaling pathway or something, or activating it less.
That's very interesting. I would like to know if the lifespan inducing effects were independent of weight. So when you look at the data, it all kind of comes down for many, to caloric restriction and weight. Not everything. I mean, like I said, the ketogenic diet Eucalorc has amazing neuroprotective anti-seizure benefits. But I would like to, and I think with the rapamycin data, I need to dig into that data. I've been wanting, and the acarbose that you mentioned, to see if these effects are independent. I think people know to look at that, but in doing this work, I realize it's very, very important to look at the caloric restriction effect, because there's a lot of studies like on EGCG. I got very into like turmeric and EGCG and all these things, which do have benefits. But a lot of the benefits was, when you mix up some of these things in the food, it becomes less palatable and the animals just eat less. So they tend to lose a little bit of weight. And then you get a robust effect from that. So, but I'm very interested in looking at that. So I'll look at his work and dig into that, and trying to remember the question that you had leading up to this. I
was asking you about the insulin signaling pathway in ketogenic diets.
Oh, okay, yeah, insulin. So that's how we actually make ketones, through suppressing insulin. If you're on a ketogenic diet and you're in a state of ketosis, or if you're fasting for a week and you're in a state of ketosis and someone injects insulin in you, it actually rapidly shuts off ketogenesis.
Hmm.
So that's a counter-regulatory effect. Actually, when we're in a state of ketosis with a diet, we eliminate some ketones through ketonuria, and then as ketone levels get elevated, they tend to cause a little bit of a release of insulin, and then that sort of turns down, not off, like kind of like a rheostat. It kind of turns down hepatic beta oxidation of fatty acids, which then decreases ketogenesis. So the mechanism of the ketogenic diet, is a big part of it is suppressing insulin, IGF-1, mTOR, and then by virtue of insulin, glycolysis. So glycolysis, hexokinase and other enzymes are kind of, regulated by insulin. So glycolysis tends to go down, and then you have a decrease in ATP and ATP-activated potassium channels, which can change the neuropharmacology of the brain. There's people who study that who are convinced that it's glycolysis inhibition. There's actually a drug called 2-deoxyglucose, which is like considered a ketogenic diet in a drug, in a pill. And then that's been used for epilepsy as a drug that mimics the ketogenic diet simply by restricting glycolysis. So, and that's through the insulin inhibition of insulin pathway. So that's the fact that 2-deoxyglucose works and has anti-seizure effect is kind of good support that the ketogenic diet is working through suppressing insulin signaling.
Yeah, no, that's very fascinating.
Mhm.
I hope there are more studies on that, but also your
Mhm.
point on, a lot of these studies, it might come down to caloric restriction. That's a really
Yep.
good point, because I'm sure, I'm not sure if you've looked into the rapamycin data, if any of the studies actually end up doing that, whether maybe some of the effects are due to caloric restriction as well, because I believe that's the case with fasting too, where people sometimes, when people say fasting has beneficial effects, sometimes it really
Mhm.
just comes down to caloric restriction. And
Mhm.
I'm sure maybe fasting has benefits independent of that, but as far as I know, I don't think we have good data on that.
Yeah, it's a good question. I just interviewed Dr. Sachin Panda, and we
Hmm.
had this discussion. So we're hosting a conference, the Metabolic Health Summit. Don't mean to plug it, but he's a speaker, and we have Dr. Jung Rowe, and Rhonda Patrick is also a keynote speaker at this event. So we've had these discussions with all of them, actually, the effect of calorie restriction and time restricted feeding. So are the benefits of time restricted feeding, i.e. intermittent fasting, completely independent of calorie restriction? I would say absolutely no. You get a lot of the benefits. When you're eating within a compressed window, you inadvertently eat less calories, or at least most people do. And if you couple time restricted feeding with a lower carbohydrate diet or a ketogenic diet, then it tends to, I think, magnify the effects. But he made some good points when I was asking him this question. There are many benefits that you get with like synchronizing your circadian rhythm by eating within a certain time window, and many other benefits independent of calorie restriction that occur with that. But I really think that the main benefit of intermittent fasting, time restricted feeding, is that it becomes a tool for us to reduce our calories, to create a calorie deficit. Is probably the main benefit of eating that way.
Yeah, no, absolutely. I'm sure there might be other benefits too, and we'll find out more.
Yeah,
There's
there undoubtedly are, but I think it's
Yeah.
like, a lot of people argue calories in, calories out, or is, it's sort of that. I'm in the camp that calories matter 80%, but that 20% that's independent of calories, that's very important too. And I think we need to understand that. And then I think we need to acknowledge and appreciate that many of these types of diets, whether it be a vegetarian diet, vegan diet, ketogenic diet or time restricted feeding have remarkable effects at, reducing and helping to promote calorie restriction. It's easy to lose weight, generally speaking, but sustaining that calorie deficit becomes the problem. And if you are the type of person who's going to do intermittent fasting, then that can work for, or the type of person that can restrict carbohydrates, then that becomes a sustainable way for you to moderate your calories. And this is important to know. That's why not everybody, there's different methods for different people. And I think you have to screen people. In our clinical trial, we have to do a lot of heavy screening before we get people into the clinical trial to understand their food preferences, their lifestyle, and things like that.
And on the point of calories and composition of food, this reminds me of your conversation with Lane Norton on Joe Rogan's podcast.
Mm-hmm.
And I think he was trying to make the point that, if someone's into bodybuilding and working out a lot, if they have, say, 1 or 2 Pop-Tarts, it's still fine. Whereas I think you made the point that, sure, it could be fine, but what's necessarily good for bodybuilding isn't necessarily good for health and longevity purposes. And I do think, I agree with you here more that sure, like you can reduce calories, probably still eating Pop-Tarts, but what are the what are the detrimental effects of that spike in sugar or glucose that you're getting there?
Yeah, he brings up a good argument. He tends to let the people who are most interested in ketogenic diets tend to be a fitness community who do not need to follow a ketogenic diet. So they're kind of like interested more or less in body composition alterations and staying lean and fit and looking a certain way from an aesthetic point of view. And there is something to the idea that body composition is a huge factor in longevity. So, and I just had this conversation with someone. You can look at all the different biomarkers, but simply taking a picture of yourself, before a diet and during a diet, I can look at pictures of someone sort of that's a heavy person before a diet and see like a time-dependent pictures over like 12 weeks. And I could predict like the changes that you'll see in their triglycerides or high sensitivity C-reactive protein, their insulin and things like that, just by looking at pictures. So I think there's something to this effect. It's like, don't worry about, losing fat. Don't worry about what you weigh. Just pay attention to eating good. But I think it is important to, I'm of the opinion that you want to maximize the amount of muscle you have and strength. And if you can lose weight and maintain your strength, that's really the key, because strength and muscle is so important. Muscle is an endocrine organ that releases myokines and hormones that have longevity effects. And it's part of our independence as we age, age-related sarcopenia. And the more muscle and strength we have will correlate to higher bone density. So and you have the time to build muscle is now, especially for women. It's probably more important for women to do strength training now to build that bone density, because you want to develop metabolic currency in the form of as much skeletal muscle and bone density as you can build now, because that's going to pay big dividends as we age later on. So I'm a huge fan. I wish I did more research on like weight training and things like that, but I want to add that to, it's hard to get, mice to do deadlifts and squats and things like that. But simply, you don't have to do that. Like simply running on a treadmill and stuff, just exercising and getting your body moving can have big translatable effects.
Yeah, I'm a huge proponent for weight training too. But when you say especially for women, is it that women lose muscle mass faster? Or I guess it's harder for women to gain muscle mass, I believe. But is it, are they losing it faster too?
Good question. I think males and females lose muscle at around the same weight or around the same kind of time scale. With males, they have a sharp dip in their testosterone, like in their 40s. And then the lowering of testosterone can lead to like more rapid muscle loss. And with women, I haven't really delved into that literature that much, but I do know, generally speaking, it's super important for both males and females. But I think of bone density because in men, you don't really hear too much about, osteopenia and, and bone loss. But in females who do, and females are told to just, hey, take a calcium supplement and then that'll, build your bones. But no, it's actually putting stress on the bone, doing weight-bearing exercises, and the bigger muscles, the larger muscles we have, that actually puts tension on the bone as we lift things. So bigger muscles correlate, not all the time, but most of the time, to higher bone density. So doing weight-bearing exercises and ensuring, your vitamin D levels are sufficient and you're getting sufficient calcium, and the soil is pretty depleted in calcium. So just saw a whole lecture on this, how we should be taking calcium supplements. And I take a beta hydroxybutyrate supplement that's kind of, bound with calcium. So it's like, after looking at that, but we live on a farm and I've been meaning to test the soil to see the calcium levels on that. But I think if, you get your nutrition on and you supplement and you're lifting weights, you're building the bone density, that's pretty much all we can do. There's a lot of drugs that are being studied to increase bone density too, but these will probably have side effects. NASA's looking at this because in zero gravity, astronauts lose a lot of bone density. And that's a big project within NASA, actually, to test these drugs. And then, I think they have translation to humans too. I think eventually a lot of women will be put onto drugs to increase bone density. But I think they could make out much better just lifting weights.
I agree. Yeah. I feel like sometimes a lot of the answers come down to just exercise more and lift, which is funny.
Hey everyone, if you enjoyed today's episode, then help me out and share it with a friend or two. If you want to stay tuned for upcoming podcast episodes and find all the previous ones, sign up on my website, livelongerworld.com. Lastly, you can also find me on Twitter, where my handle is @livelongerworld. That's it for now. Stay in good health, and I'll see you next time.